Hello everyone, welcome back to another class on regeneration biology, and today's topic name is xenotransplantation versus organ culture in practice. So we will try to understand what xenotransplantation is and what is the scenario in which that is needed, and what are the different angles and aspects of organ culture in practice. So xenotransplantation and organ culture, they go hand in hand. Okay. So in the sense, like if I want to give a simple example, like ordering food online versus cooking, something like that. So given a scenario, anyone will order food online so that you get the food without any difficulty. But if no service is there, it is band, or no restaurants are there or something, that is the time where you have to make food of your own and you can have it. And one can say it is less tasty, or one can say, oh, it is less tasty but it is more quality, because ordered food, I don't know how it is made, who made it, and was it made fresh or old, except that it is hot, we have no idea. But when you make it, you have a good control of your lot of QC, something like that. So if X doesn't work, you do Y. So that's how xenotransplantation and organ culture go. Xenotransplantation is the transplanting of living cells, tissues, or organs between different species. That means you take from pig to human, or you take from rat to mouse, or rabbit to elephant. This is xenotransplantation. Organ culture, the process of cultivating and maintaining organs or tissues in the laboratory setting, is a field with significant potential to address the global shortage of transplantable organs. Transplantable organ can be from a donor, or transplantable organs can be from another species. So xenotransplantation. So when this doesn't meet the demand, then comes the organ culture. While xenotransplantation aims to provide new source of organs from animals like pigs, organ culture offers a way to regenerate or repair damaged organs in vivo. Essentially, both does the same job, but in reality, the xenotransplantation is definitely not a suitable solution. You can see in a short while. Both approaches are actively being researched and developed with AI. These days we use a lot of artificial intelligence tool also play a role in enhancing their effectiveness and safety. In the sense, sometimes you have to assess, can this person be given immunosuppressants so as to retain this xenotransplanted organ. Sometimes the immunosuppressant will make the person more vulnerable, they can die of infection. So you have to take a decision. Xenotransplantation, its potential shows that the xenotransplantation, xeno means strange, and transplantation means, putting one organ to another. Xenotransplantation could significantly increase the availability of organs for transplantation by utilizing animals like pig, which have organs relatively similar to that of human organs in terms of its size and functionality. And the challenges include, significant challenges still remain, including overcoming immune rejection and ensuring the safety of animal derived organs and addressing ethical and religious concerns. Sometimes it may happen from an animal, when you take an organ, it has some virus, but it was harmless in that organism. But once you get it, maybe that virus will kill you. You would have lived some more years with that defective organ. The moment you transplant it, that virus present in that xenotransplanted organ can kill you. So this is what one should be bothered about. And it also, a lot of tweaking has happened because of the artificial intelligence. It is being used to optimize the process of creating genetically modified pigs, which reduced immunogenicity and predict and manage the rejections risk and improve the xenotransplantation procedures efficiency. These days it's a land of artificial intelligence, so that can handle so many parameters and come up with a logical conclusion that which is the best way that can be approached, or that can be adopted for treating this patient. So one example include CRISPR-Cas9 gene editing is being used to remove immunogenic molecules from the pig organs, making them more compatible with the human recipients. We usually refer to this as humanized. If there are some 5 proteins that are present which can be immunogenic in human, if you take that organ and put it. So what if in pig itself you get rid of those proteins by editing or mutating those proteins, and then you take that organ, then they will never be present expressed, and hence your immune system will not reject that pig organ. So the public perception about this kind of approaches and the concerns about xenotransplantation includes some are ethical, religious and social factors, and that need to be addressed for implementing it. Organ culture, on the other hand, it has a huge potential. That is, organ culture offers the possibility of regenerating damaged organs for creating artificial organs to replace diseased or damaged ones. So that is the simple approach of organ culture. The challenges include maintaining organ viability and functionality in vitro, creating biocompatible scaffolds for organ regeneration, and addressing the challenges of vascularization and innervation are the major hurdles. Say you put an organ, but if that organ is not getting the blood supply from the host, or it's not getting the nervous tissue command from the host, then that is not going to function the way it should, because each and every organ in your body is functioning because of the inputs coming from your nervous system. So your nervous system should be able to control that organ. Then only it will become a part of the life. And here also artificial intelligence plays a major role. AI can be used to analyze intricate processes of organ regeneration, optimize culture conditions, and to design biocompatible 3D printed organ scaffolds. So all these angles, the AI can come into picture. Some example, AI can be used to predict the growth and development of cells in vitro, leading to more effective organ regeneration strategies. If you have a given cell or tissue in your hand, you can predict what is the best medium that can be used, how fast it can grow, etc., based on lot of other parameters that is available to analyze for the AI. And the ethical consideration also comes into organ culture. The ethical implications of creating artificial organs and the potential for its misuse must be carefully considered, because it can, like any technology, can have its side effects, that is misuse. Evolving areas in heart transplantation. We'll take heart as an example because one of the major organ that is being looked into for organ transplantation. Xenotransplantation refers to grafting of or transplanting of organs or tissues between members of different species. This we have seen it. With increasing numbers of patients with end stage heart failure and shortage of donors, which means heart donors, there has been renewed interest in the possibility of cardiac xenotransplantation. Like we discussed, you can make a humanized pig heart and you can transplant. Advantages include unlimited supply. No one will say that, oh, pigs are in short supply, hence we cannot have enough heart. So animal, we can grow them as many as you want, unlimited supply, and elective availability of donors, organs, expansion of candidate pool. Sometimes you cannot choose, oh, I want only 25-year-old person's heart because I don't want someone who is 50-year-old heart. You cannot choose. But whereas if it is an animal, you can choose the age, what should be the age based on your demand. And you can also expand the candidates pool, and avoidance of detrimental effects of brain death and donor organ function. This can be avoided. Although monkeys and baboons are most phylogenetically similar to humans, given the ecological and ethical restrictions, the pig is the only animal available to replace human tissues. In various other angles, the pig is much closer to that of human. The pig physiologically similar to humans and organ size is compatible. It can be raised in controlled environment and are more importantly genetically modified, can be genetically modified so that there will not be too much of immune rejection from the human body. So the main limitations of xenotransplantation are the rejection, which includes hyperacute and acute and chronic vascular diseases, and the risk of transmitting zoonosis, that is say various diseases called zoonotic diseases, like many viruses that is present in the host, in that donor can come into the host, and it can create some zoonosis. So the development of genetically modified pigs has allowed the expression of human antigens that can reduce the risk of rejection and provide hope for a future feasibility. That is called a humanized pig or humanized organ. Xenotransplantation, if you look into this table taken from this publication, you can see the current the scorecard of the xenotransplantation, advantages and disadvantages. Advantages: unlimited organ supply and elective availability, expansion of candidate pool, avoid effects of brain death on cardiac function, potential for genetic manipulation, cost saving, etc. Advantages. Disadvantages include size mismatch, ethical restrictions on primate use, ecological restrictions on a given species use, hyperacute, acute and chronic vascular rejection, coagulation dysfunction, transmission of zoonosis, that is zoonotic diseases. These are all the limitations. This is a schematic heart transplantation status. So this is a central illustration, and it evolves areas of heart transplantation. The central illustration demonstrates three broad areas that is to be discussed. Area number one is the novel immunosuppressive agents to treat allograft rejection. That means your T cell mediated response is the one which is killing the transplanted organ, attacking. So can you selectively kill rather than broadly suppressing the immunity? And new modalities in imaging and the genomic medicine to screen for the rejection possibility of, like we mentioned about the AI, you can predict the possibility of a rejection can be calculated or evaluated beforehand. And the future potential ways to increase the donor pool, that you should be able to choose. Like you're going to buy a shoes, you don't buy, okay, 8 number is my shoe, or give me any 8 number. It will, no, sometimes you have to wear and feel it. Is it okay, comfortable? So when donor pool is more, you will be able to choose the best. Novel immunosuppression is one approach. Different drugs act in different ways to protect the transplanted organ. Screening for the possibility of rejection that can be done, and potential for increasing the donor pool. That is what the three different cartoons that has been mentioned here. So the stem cell therapy and tissue engineering is another promising approach. So the landmark scientific work has demonstrated that the resident cardiomyocytes can self-renew. The annual cardiomyocyte turnover is around 1% in the young adult, and it decreases to 0.5% in elderly people. So promoting this endogenous repair process, your heart is being repaired on a small scale, just like your skin is getting repaired, but not in an adequate level. Promoting this endogenous repair processes can enhance the regenerative capacity and modify or reverse the adverse LV remodeling that is occurring because of the cardiac malfunction or blood supply is cut, etc. Regenerative cell-based therapies ultimately aim to restore normal myocardial function through both direct cell-mediated and indirect paracrine-mediated repair mechanism. Sometimes you can stimulate that area with some growth factors, that is paracrine factor, or you can have the cells, cardiomyocytes derived stem cell, and deliver in that area so that the damaged area can be fixed. Initial trials utilized the first generation cell-based therapies, predominantly consisting of unselected progenitor cells, mesenchymal stem cells, and endothelial progenitor cells taken from the bone marrow. Although they seem easy, they carry some baggage. However, these populations may not differentiate into cardiac tissues despite having the potential to differentiate into cell types within the myocardium. Two problems. One is it may not effectively differentiate, or even if it effectively differentiated, there is a possibility that it may change into something else because it originally derived from the not from the heart but from the bone marrow. So this is the main issue. So the next generation therapies include the isolation and expansion of the cardiac resident stem cell, or the creation of cardiopoietic stem cell, not from bone marrow or any other tissues, through the lineage specific differentiation of the bone marrow derived progenitors, commonly called as cardiopoiesis. So you, even if your source can be anything, you first make the stem cells that is specific to the cardiac tissue. So this is the first step, and then there is less likely that it will change its identity. Cardiopoietic stem cell therapies have demonstrated the promise of proof of concept studies, including C-Cure, and it's a cardiopoietic stem cell therapy in heart failure. That is the full form trial. So cardiac tissue engineering is another novel approach to regenerative therapy that uses engineered human myocardium, that is EHM. The challenges of EHM is to create functional cardiomyocytes with the electromechanical properties of human myocardium. A recent study optimized the cellular chemical components that can be used for the EHM generation, that is engineered human myocardium, resulting in an advanced cardiomyocyte maturation. So researchers also have transferred the EHM into the nude rats. Nude rats means the immunity is compromised, using epicardial approach, leading to revascularization of the grafts. So you took the cells, grafted onto a recipient nude rat, and these cells got colonized and differentiated, and they got revascularized. That means the blood supply is restored. However, the electromechanical coupling to normal myocardium was not clearly seen. That means the contraction is needed, not that just a structure. That was not properly coupled, and that was one of the drawbacks. Although not yet ready for clinical trials, EHM is a promising approach to cardiac repair that may soon be feasible. So it's a question of time. You have to standardize it. Can I get it into electrocoupled also effectively? So now comes the total artificial heart. The development of total artificial heart, known as TAH, to replace the failing heart has been the focus of global research for several decades. The first successful use of TAH was done by Dr. Denton Cooley in 1969 in a patient bridged to heart transplant. You have also connected the patient with the heart transplant. The only device currently approved by US Food and Drug Administration, that is a 70cc SynCardia temporary total artificial heart, that is from a company in Arizona. The THT is currently indicated as a bridge to organ transplantation in patients with the serious heart issues. Means it is not a permanent solution, rather a makeshift solution until the organ transplantation is done. So this is a picture of the total cardiac replacement for irreversible biventricular heart failure. This is the instrument that is used, that is Karma total artificial heart, that you can see TAH in the panel A, and it is been used. It has got two valves, prosthetic valves, and also it has got sensors to detect it, and also it can prevent the blood clotting formation, etc. And this is another version, that is by Vacorta. That is another approach. The advantages include magnetic bearings, and it's compact in size, and it's a single rotational component. Several mechanical properties are there, so either of them can be used as a artificial heart. So from here, if you conclude, it has been 15 years since Dr. Christian Bernard performed the heart transplantation in human-to-human transplantation back in South Africa. And its advances in immunosuppression, donor and recipient selection, surgical techniques, and post-transplant care all have improved over the years, and it is made into an excellent long-term outcomes are established these days. However, significant barriers remain still, including early and late risks of organ rejection and vasculopathy. Like sometimes organ will be rejected immediately, and if it did not reject, you can go smoothly for a few years, and that is not the end of the show, because you can get a rejection much later. So you can't be tension free. You cannot be tension free at all. And it can also bring in the risk of infection and malignancy because of immunosuppressors you are taking. Although the donor pool is cautiously being expanded, it is unlikely to fill the needs of continuously growing sick patient population. More hearts are needed. So the future depends on the progress of stem cell research and tissue engineering, and the progress in the xenotransplantation, while optimizing the post-transplant health through the improved immunosuppression strategies, and acute and chronic rejection could be avoided. So the reversal of the heart failure to avoid the need for organ transplantation is also as important as the organ culture itself. So human organ culture. In human studies, the critical for developing new diagnostics and therapeutics are limited by ethical and logistical issues. And the preclinical animal studies are often poor predictors of human responses. That is also another problem. And the standard human cell culture can address some of these concerns, but the absence of normal tissue microenvironment can alter the cellular responses. Hence, you may not end up getting the same organoid or whatever you are looking for. Three-dimensional culture, that is also known as 3D culture, that positions the cells on synthetic matrices or organoid or organ on a chip cultures, in which different cells spontaneously organize and contacts with the other cells in a natural matrix, only partly help in overcoming these limitations. So human organoid cultures known as HOCs are more faithfully preserve the in vivo tissue architecture and to better represent the disease associated changes. So it can help in much better way, HOCs. And HOCs can be combined with the traditional and more modern morphological techniques to reveal how anatomic location can alter the cellular responses at a molecular level and permit the comparison of different cell types and shapes. Because if an organ is put in place, it should be supported by the blood flow and blood pressure, etc. Sometimes if the pressure is high in a given area, the organ may not sustain. So this also one has to take care of. HOCs are the prominent not only just for transplantation, it's also very prominent in studying inflammation, cancer, and stem cell biology itself. HOCs, how it is used in organ transplantation. In normal kidneys, ECs of glomerular, one example from the kidney, glomerular and peritubular capillaries, PTCs, express higher level of TNF receptor 1, that is tumor necrosis factor receptor 1, associated with inactive apoptosis signal regulating kinase known as ASK1, and the absence of activated ASK1. So these are all scenarios that you can normally see in normal kidneys. On the renal canal, the types of both TNFR1, ASK1 and TNRF2 is only minimally expressed in any cell type in a given normal kidney. So this expression pattern of TNFRs differentiates the renal cells in situ from the cultured renal cells. That is very important, because when you culture, if these are not there, receptors are not there, they cannot mimic a normal renal tissue, where both receptors are typically expressed in the same cell. It also differs from the patterns of rejecting renal allografts or ischemia perfusion injury, and where the resident renal cells lose TNFR1 expression and the ECs of the PTCs and tubular epithelial cells express the TNFR2. Effects of TNF signaling, that is tumor necrosis factor signaling, on different renal populations can be analyzed in the organ culture, because if these receptors are expressed, it is very easy to analyze. Treatment of normal kidney organ culture with the TNFR1 selective mutein results in the activation of ASK1 and the induction of cell death in the ECs, and also the glomerular and the PTCs, while the TNFR2 selective mutein upregulates the TNFR2, results in the phosphorylation of endothelial to epithelial tyrosine kinase, and it initiates the cell cycle entry of these ECs of the PTCs and also present in the TECs. So TNF does not affect the expression of either receptor in the cultured cells. That is because of the unique nature of the culturing condition. It has to be induced if you want to put it into the host. So if you look at comparison of different culturing methods, 3D cultures is a wide range of 3D in vitro models is emerging to mimic human physiology better, and at the same time to reduce the animal experiments. 3D cultures use different types of porous scaffolds derived from the naturally engineered substrates, and gene expression profiles of cancer cells often differ in the cells that is grown in 3D as compared to the 2D cultures. In particular, genes involved in angiogenesis, proliferation, invasion, migration, and chemosensitivity. The 3D cultures have been used to determine the effects of cytokines and cells seeded into 3D cardiac ECM scaffold that have been shown to increased calcium signaling and kinetics, that is promoting stem cell maturation. So, but we should understand that the contrast to HOC, human organ culture, ECM scaffolds used in 3D cultures fail to emulate the natural biochemical and physical properties of the ECM recognized as independent factors that influence the cell activity. So furthermore, we should note that the basement membrane derived scaffolds may contain undesired components like viruses and growth factors. So if you look further, other matrices permit cell attachment but not easy detachment of cell mimicking assay development is difficult. And even with the generation of 3D biological scaffolds via organ decellularization, which we discussed a few classes before, current system failed to offer complex in vivo tissue vasculature for the supply of nutrients and oxygenation, as well as for the removal of the waste material necessary for promoting attachment, differentiation, and the proliferation of the cells. Moreover, the clonal variation across various strains of cell lines, 3D culture techniques used in different laboratories may also influence this kind of experiments. Unlike the human organ culture, cell lines used in 3D cultures lose many of their native in vivo characteristics once removed from the primary tissue, because they are known for changing their gene expression profile once you put them into culture. Most recent research have shown that in vitro models ignores the origin of cell lines and histological characteristics because of the unavailability of this data, that is the gene expression data. For example, one gene, that is SKOV3, an epithelial ovarian cancer cell line that is widely used in 3D in vitro model for studying the ovarian cancer, that bears a striking histological resemblance to a clear cell ovarian cancer, which is the least common histological subtype for an invasive disease. So some situation, cultured scenario also, it will mimic exactly like the normal scenario. So the 3D bioprinting, the system incorporates biology and tissue engineering to develop biological substrates and restore and improve and maintain the tissue function. The three most common 3D bioprinting mechanisms, which we discussed in detail in the previous class, include the inkjet and also laser and also extrusion bioprinting. You can refer back our previous class. So this system offers additional biocompatibility and capacity for uniform cell incorporation. This system has shown to promise in gene and drug delivery with the precise placement during tissue construction. However, compared to human organ culture, HOCs, the limitations of this system include difficulties in managing single cells, overdrying leading to the failure of biological systems, and cell damage and altered phenotype caused by the printing processes itself. Bioprinting requires fine-tuning matrices for optimal simulation conditions and deposition of cells and scaffolds. So, thus, the precise delivery of cells and biological factors to the designed 3D cultures remains to be unresolved. So if you look into the organoids, the system stands at the forefront of 3D approaches as the more accurately recapitulation of the in vivo characteristics of the original individual tissue, because of the ECM and cell-cell interactions are intrinsic in nature to the culture. Organoids have been successfully generated from healthy and diseased human tissues and pluripotent stem cell. Organoids from the primary tissue sustain basement membrane extract that are hallmarks of the original tissue in terms of its architecture, cell type composition, cell renewal properties, etc. Patient-specific organoids have allowed functional genomic studies which can be done to diagnose the future. However, in comparison to HOCs, organoids lack the histological diversity and sequential differentiation which occurs in adult tissues. So, lot of secondary alternatives also has to be taken into consideration. To conclude, in contrast, Other experimental cultures commonly employed for basic research, clinical drug testing, human system, HOCs have got their unique place. And also, the increasing data also suggests that they exhibit reduced or altered key functions also can be seen. HOCs preserve the integrity of the cells and matrices during organ culture, and HOCs can be modeled in a wide range of applications that can permit many functional mechanistic studies. We will study more about the organ regeneration in the next class. Thank you.